Welcome back. We'll continue our lecture series about energy production. So remember we talked about glycolysis, and that was the first step. Right. First step in cellular respiration, what we said was the catabolic breakdown of glucose. All right. So remember that this right here so that was anaerobic, right? Which meant without oxygen. Okay. So usually this will produce our NADH, which are electron carriers, right? I got 2 ATP out of it, net, and 2 pyruvate. Now those pyruvates I'll send to the mitochondria, right? And that mitochondria will produce the bulk of the ATP for me, right? So, and then this, we're going to see is aerobic. That means it requires oxygen, okay? So we have all that going on, all right? All right, so all of that process requires that I have oxygen. So to go to the next step after glycolysis, I need oxygen, right? But if I don't have oxygen, so if I don't have enough oxygen, right, I start to do fermentation. So fermentation is the process without O2, right? So that's my process without O2. So when I have my fermentation, so yeast, things like yeast, right? Remember, all things do glycolysis, right? Yeast produce ATP in the absence of oxygen. They don't have lungs, they don't breathe, right? So it starts with glycolysis. So all stuff starts with glycolysis, right? So glycolysis is the same. They produce 2 ATP, net, 2 NADH, and 2 pyruvate. So I start with glucose, right? I use some ATP and I make ATP. So this is the end. So right here is the end of normal glycolysis, right? So I've produced my NADHs, I've produced my net ATPs, but for yeast, right, they need to recycle those electron carriers, right? So they'll continue the process and they'll produce ethanol, right? So ethanol is an alcohol, right, used in like beer. So beer uses yeast to produce the ethanol, right? And as long as I keep feeding the yeast glucose, right, and I remove the ethanol because it'll become poison to the yeast eventually, they'll keep, making more ethanol, right? So here's a big difference in glycolysis, right? No CO2. So in your body, you start with glucose, which is a 6-carbon sugar. I make pyruvate, which are 2 3-carbon sugars, so I still have all the carbon. The difference here is in fermentation, 2 CO2 are released. That's why I use yeast to make bread, right? Because the CO2 makes the bread fluffy, right? Because they're making alcohol which burns off, and the CO2 is trapped in that dough, and it makes the dough rise. Okay, so CO2 is released, and ethanol is C2H5OH. Right, so that's the formula for ethanol, right? So instead of 3 carbons like pyruvate, I only have 2 carbons here. Right? So things like yeast will do alcoholic fermentation. You're not yeast though. So in the absence of alcohol or absence of oxygen, you'll do lactic acid fermentation, right? Same thing. It starts with glycolysis. You'll get your 2 ATP net, your 2 NADH net, and your 2 pyruvate, right? So down here, down to this process, same thing. right? But you will then recycle your electron carriers, right, and produce lactate, otherwise known as lactic acid. So when you exercise, you use your stores of oxygen really quickly, right? And you can use that oxygen to make ATP, but very quickly you start to go to anaerobic respiration and you start to store lactate or lactic acid, right? And the purpose of that is when O2 is available, it can be converted back. Right, so when O2 is available, I can feed it back into the cycle, right, and still make energy out of it. So I'm just kind of saving it for later, but I'm getting the 2 ATP molecules that I can use right now, which is not a lot, right? So make sure you know, because this ends up being a test question all the time, right? It's performed in our muscles, and it's anaerobic, right? So when you run out of oxygen source, if you're a sprinter and you run the 100 meters, right, you have run out of oxygen before you hit the end of that 100, right? So about 60 meters in, you're completely out, right? And then you're just depending on lactic acid fermentation to get you to the finish line, right? That's why you can't sprint for a full 100. You can't sprint for a mile, right? You've got to slow the process down and let your body start bringing the oxygen back into the system, and you're switching back between fermentation and cell respiration. Right, so we'll continue with cell respiration now. Right, so we'll talk about the Krebs cycle. So we had glucose. Right, so this is 6 carbon. Right, we went to pyruvate, 2 of them, which are 3 carbons. Right, so at this point I haven't lost any carbon. All right, and this process right here was glycolysis. And this was a cytosol. Right, so that's right here. Right, so I've got my 3 carbon sugar right there. Right, so the next thing is so this now this can be transferred to the mitochondria. Right, so the first thing that's going to happen, right? So the Krebs cycle is also known as the citric acid cycle. I forgot to tell you. Right, so 2 molecules of pyruvate enter right through active transport. So it's going to require ATP. So the ATP that was made in glycolysis is going to be used to move the pyruvate into the mitochondria, right? So the first thing that's going to happen is you're going to change the pyruvate, right? It's going to be transformed to acetyl coenzyme A, right? So this is a 2 carbon sugar, right? And then CO2 is released. So I'm going to take my first CO2 off, right? My first CO2 is going to come off as my pyruvate, right? Goes into the mitochondria, and then I'm going to release a CO2, right? So this CO2 like this. And I'm going to make another electron carrier, right, an ADH. But remember I have 2 pyruvates, so I get 2 acetyl coenzyme A, so I get 2 CO2s and 2 more NADHs, right? So that's our process. That's where we're beginning, okay? So, and from there I can start the Krebs cycle, right? You can see right here, the Krebs cycle makes more electron carriers, right? It makes more ATP. It makes FADH2, which is another electron carrier we'll talk about, and the coenzyme A. Remember, enzymes aren't used, will be recycled, and it will be started over again. Okay. All right. So acetylcoenzyme A, we'll talk about that a little bit. Let's switch back to red. All right. So before the Krebs cycle can begin, right? Remember, I have to make that coenzyme A. Remember that the first CO2 is produced. Right now we have acetate. Right. So if you see acetate has 2 carbons. Right. We talked about that, and that's why it's called the citric acid cycle, because of the acetate. Right. Coenzyme A, right, is added to the molecule. So here's essentially what we did. We had our 2-pyruvate, right? From that we made 2 CO2, right? 2 NADHs and 2 acetylcoenzyme A's, right? So 2 molecules of CO2 are produced. So I started with 6 carbons, and now I have 4 carbons left. Right? You have to remember your carbons, because it will be important to count where your carbons are later on. All right, so those are all going to be fed into the citric acid cycle, right? So the coenzyme A's, right, so these are fed into the citric acid cycle, otherwise known as Krebs, right? Because Krebs is the guy who described it, right? So he gets that name. But then in college, they'll start to call it the citric acid cycle for you. All right, so it's important to remember, so this is a cycle. So if you can see with the cycle, where we start is where we end. And then it just keeps on going. So your mitochondria, remember the mitochondria? They produce ATP. So this cycle is always running. As long as there's pyruvate, it's going to feed it in there, and it's going to keep going, right? So as long as we feed the mitochondria, it's going to stay happy, and it's going to keep working for us. All right, so here we go. So step 1. Let me make this a little bigger for you so you can see it. All right, let me erase this because I don't want those. All right, so step 1. So I get my acetylcoenzyme A. Remember, that was made in the mitochondria, and it's fed in. And then we get citrate. So citrate is the first molecule. That's why it's called the citric acid cycle, right? And so from that, I make an NADH. Let's count them. So there's 1 NADH. I'll count that in green. Right? So I have oxaloacetate. So here's my molecule I have to remember. So oxaloacetate will join with my acetylcoenzyme A. It'll make citrate, which is a 6-carbon sugar. Right? So it comes, and I take and I release the CO2. So CO2, there's my first CO2 right there. I make another NADH. I release another CO2. Right? I make a GTP. It's kind of like ATP, but it uses guanosine. I make a FADH2. Make another NADH. And then I have oxaloacetate again, and it can accept another acetylcoenzyme A. So let's just know the picture. So here, so NADH is produced, right? So I start with acetylcoenzyme A. Let me erase this, and I'll make you a better list. Let me raise this up a little bit. All right, so I start with acetylcoenzyme A. All right. So this joins. It joins with oxaloacetate to form citrate, and then I start making stuff. I make 3 NADHs. make 2 CO2s. I make 1, right? And I make 1 GTP, right? So remember, so these 2 CO2s, right? Remember my acetyl coenzyme had 2 carbons, so I've used both of those carbons, so I got the energy in both of those, right? Remember, this is going to be times 2 because I have to feed 2 acetyl coenzymes in, 1 at a time. So I'll actually get 6 of these. I'll release 4 of these. I'll get 2 of these. And I'll get 2 of those. And this is sometimes called ATP as well, because I can use that GTP to make more ATP if I have to. Right? So that's my total. So these are my totals. And then my coenzyme A, remember, gets fed back in to make more acetyl coenzyme A from pyruvate. Right, so I release 4 CO2s here, right, total, and I release 2 when I change pyruvate to acetyl coenzyme A. So that's our summary here, okay? So again, I start with glycolysis right here, right? I convert that, so this is in the cytosol, right? I move it to the mitochondria right here. So this is all going to be in the mitochondria. Right, so the first thing I do is I make acetyl coenzyme A. Then I fit it into the cycle, right? And I start to produce CO2, NADH, ATP, it said GTP, because that's what we'll say in college, FADH2, which is another electron carrier, and then the cycle just continues again. All right, so that's where we are today so far, and I'll talk to you soon.